This is BeerSmith episode number 114, and Dr. Pat Hayes, professor of barley breeding from Oregon State University, joins me this week to discuss barley. Thank you to this week's sponsor, Craft Beer and Brewing Magazine. They're now offering a full 6 issues a year, up from 4 at a great discount. They're offering a new discount code, which gives you 15% off everything they sell, including subscriptions and training. The new code is BeerSmith 2015. I encourage you to check out this great new magazine for homebrewers at beerandbrewing.com and use the offer code BeerSmith 2015 to get your 15% discount today. And also, BeerSmith. Design great beer, brew with confidence, and brew your best beer ever with BeerSmith brewing software. BeerSmith lets you create your own recipes from scratch or access tens of thousands of recipes found on the BeerSmith cloud. Also, our desktop and mobile version work together seamlessly to make brewing easier. Give BeerSmith a try today by downloading the free 21-day trial from BeerSmith.com. Get your free trial today at BeerSmith.com. And now let's jump into this week's episode. Today on the show, I welcome Dr. Pat Hayes, professor at Oregon State University. Pat specialized in genetics and breeding of barley products. His recent research has been in developing cold-tolerant, disease-resistant tworow barleys. Pat, it's great to have you on the show.
Well, thanks so much, Brad. Good to be here.
So you teach courses in plant genetics, and you focus most of your research around barley. What makes barley such an interesting plant?
Well, we're so lucky to just have this tremendous history with barley. It was probably one of the first crops to be domesticated. You've got this cultural importance in barley, the whole brewing tradition, and then there's also a culinary tradition in barley, which is something that's just kind of coming back onto the radar because of some of the health attributes that barley has as a human food. And it's also a great genetics model system. That is, that it's kind of the fruit fly for plant genetics, that many things that we do in research are just simpler to do on barley than they are on more genetically complicated plants such as wheat.
Interesting. So you're saying barley is one of the simpler genetic plants?
Yeah, it's what we call a diploid plant, and that means that it has 7 pairs of chromosomes, 14 chromosomes. And so if you look at wheat, then it's got 42 chromosomes. And so it's just kind of simpler to deal with 14 things than 42. Wheat essentially just has multiple copies of the same or similar gene. So another way of looking at it might be looking at a 4-cylinder versus an 8-cylinder engine.
Very cool. Well, obviously, a lot of your research has been into the study of genetics and the power of genetics to provide better plants. What can you do using genetics on a staple grain like barley?
Well, probably one of the first things we ought to do, Brad, is kind of distinguish the overlapping fields of genetics and plant breeding. Mm-hmm. And genetics is a science that tries to understand how things work, starting with the DNA. And breeding is very much about making things better. And so genetics can inform plant breeding, and plant breeding can inform genetics. But as one example of the difference between the two, if we make a cross to study the inheritance of something in a genetic analysis, we also want the bad ones, the ones that don't have the traits that we're looking for, because they help us to understand the traits that we are looking for. In breeding, on the other hand, you just select for the individuals that have the trait that you want and throw out the ones that don't.
Interesting. So obviously you're studying genetics. What makes that so different?
Well, we do both, in fact, because I'm really fortunate to have a job at a land-grant university. And so we have kind of dual assignments at land-grant universities, and one is to contribute to the fundamental body of knowledge as best we can, and that's certainly a genetics proposition typically. And then the other is to also create products that will stimulate economic development in our states, and that's where plant breeding comes in. So, it's kind of the best of all worlds because I get to explore topics in barley like low temperature tolerance or disease resistance or flavor. And at the same time, then we get to release varieties and have the satisfaction of seeing local farmers and industries and beer consumers benefiting from those.
And you actually developed several varieties yourself, I believe, right?
Yeah, well, I can't say that I did
Or
it.
played part in it, I guess.
Yeah, it's a team thing. And so we have the Barley Project here. And so we're a kind of evolving team. And there's a couple of core folks that have been here for some time. And so it's very much a collaborative effort. And then even beyond that, then we collaborate extensively with colleagues across the U.S. and throughout the world.
Now, obviously, there's been some public backlash against genetically modified organisms, GMOs, or genetically modified plants. What's the source of that backlash?
Well, we could probably do a whole separate show just on that topic. So maybe we'll kind of step back just a little bit and again, kind of look at a few definitions and then look at the state of GMO research in barley today. So it's really important to define what we mean by a GMO. And probably the most direct and simplest definition that we have today is a non-sexual transfer of genetic information. So in plant breeding and in our conventional genetic analysis, we can make a cross between two barleys and study the inheritance and understand the underlying DNA code that is responsible for the traits that we're interested in. In a transgenic situation, you would use a non-sexual system, so you use what's called a vialistic thing, a gene gun, or you infect a undifferentiated mass of cells using a bacterial system called Agrobacterium tumefaciens to get in the particular DNA that you want, and then that resulting plant is what's called a transgenic or a GMO. There's a new technique that's newer, I should say, that is certainly out there that is going to defy some definition, and that's called genome editing. And so it's a new suite of techniques that allow you to change the underlying genetic code in an organism without introducing a foreign gene. So some of these discussions get to be pretty legalistic, and it's like how many angels can dance on the head of a pin, or how many DNA molecules can you fit in a glass of beer. So we'll just leave it on the definitional thing and then kind of explore GMOs and barley as they relate to what a consumer might find in the glass today. And that is that GMO barley is strictly an academic consideration. There are, to my knowledge, no commercially available barley varieties that are used in malting or brewing. And some of that is almost an informal agreement, I think, in the malting and brewing industries that since beer is a pure and natural product, that they don't want to cloud that definition in the mind of consumers with concerns about what a GMO might be.
So that's kind of a... So you're not actually developing these, I guess, at least for commercial use, right?
Exactly. And so when I said it was an academic issue, that it is academic in the sense that GMOs are a useful way to study or to do research on, is the gene that you think is responsible for the trait really the gene that's responsible? So we've published a couple of collaborative papers on using GMO techniques to try to understand some genes that we've isolated. And so it's very useful for that. As a plant breeder at a public sector institution, the other consideration for me is simply the cost of the intellectual property. So there is so much IP associated with that technology that chances are, even if I release some barley variety that had some really, incredible attribute, that the cost of that seed would be so prohibitively high to farmers that it just would never fly.
Very cool.
The other thing that, just kind of to sort of round out the GMO thing is that there is so much naturally occurring genetic variation in barley that we haven't tapped, that's very much the focus of what we're about in our research project. And so barley's probably got 30,000 genes. We have a barley genome sequence we can refer to. We have all kinds of DNA fingerprinting tools. And so our research is really directed at understanding the naturally occurring genetic variation in barley.
So you got enough work to do already without messing with it, right?
Exactly. Yeah. And so there's just so much to do. It's like, why get into, sort of a sensitive issue with some folks, and we can just leave that alone.
So let's get back to some of your work. I guess you recently switched from research on 6-row barley to 2-row barley. I was wondering if you could first explain the difference. I don't know how many brewers really know the difference between the two.
Yeah, we'll see if, could you see my hands here?
Sure.
Okay, so we got 3 fingers up on either side. That would be a 6-row barley. So at every rachis node, botanically speaking, you've got 3 fertile kernels. A 2-row barley only has 2. so the outer two are sterile. So a two-row has 2 rows of kernels on a spike. A 6-row has 6 rows of kernels. Mm-hmm. So it's a pretty simple trait. There's a couple of modifying genes, but in essence, it's one gene that drives 2-row versus 6-row, one gene out of 30,000. And so, just to kind of get folks thinking here a little bit, I'd say that it really doesn't matter if you're malting or brewing with a 6-row barley. But there's this tremendous cultural baggage that kind of goes along with what's a 2-row and what's a 6-row. And some of that is kind of what motivated me to switch from 6-row to 2-row.
Mm-hmm. It seems like 2-row is more popular for brewing, right?
Well, it is, and I think that is very much a cultural and historical thing. And let me just back up a sec here. Sure. The wild or ancestral form of barley is 2-row. And so 6 rows were selected post-domestication, and they were favored in some areas in the world, such as North Africa, the Iberian Peninsula, and then on into parts of Central Asia, where historically the 6 rows kind of established a foothold. Meanwhile, 2 rows moved up into Europe and were selected throughout Central and Northern Europe. And as a consequence, then, you had a brewing tradition that was founded on 2 rows, and the 6 rows figured more in a feed barley tradition or in the culinary barley tradition. Now, things sort of all got mixed up in the United States, particularly where the 6-row brewing type barleys originated, and that was when the original Caucasian colonists brought in barleys. They brought in 2-row malting barleys in general. Mm-hmm. And those 2 rows proved to be exceptionally unadapted. They were susceptible to all kinds of diseases and didn't yield particularly well, and people needed beer. So the 6-row introductions that were brought into the upper Midwest just took off. They were a hit. They were adapted. And as a consequence, you saw the rise of North American 6-row. And those 6-row barleys, as sort of a collateral sort of effect, had exceptional enzymatic power. And so that's sort of where we got this kind of tradition that 6 rows were high enzyme and 2 rows tended to be higher malt extract, lower enzymes. But being only one gene out of 30,000 genes, you can have high enzyme 2 rows, low enzyme 6 rows. And really, if the maltsters and breeders would accept the resulting products, plant breeders could crank out 6 rows or 2 rows to meet anybody's specification.
And of course, most modern malts are very highly modified, right? I mean, they've got great enzymes, right?
Well, yeah, it kind of depends on the type of beer that you're brewing. And so for the adjunct user, they're going to want higher levels of enzyme and perhaps sacrifice a bit of extract, although they'd want malt extract as well, whereas your all malt brewer is going to generally prefer a lower level of enzymes, a lower level of FAN as well in their malts, just due to the nature of the brewing process.
So what are some of the things that drove you to move from 6-row barley to 2-row barley for your work?
Well, I got to confess, one of those fundamental motivators is money, right? And in my case, the money motivator was that I'd started working on 6-row barley about 30 years ago when I just started my job here. And there was this real visionary guy at Anheuser-Busch. It was Anheuser-Busch at that point. This gentleman's name was Paul Hoskins. And he had the most enviable job that he roamed the world, as best I could understand, looking for research propositions that could, in the long run, benefit the company. And so as we got to visiting, he had this idea that someday Anheuser-Busch was going to need an alternative source of supply, and at that point they were using 6-row. And he said, maybe we need to grow barley in new places and at different times. So, Pat, why don't you work on winter 6-row barley? And we signed our first grant agreement on a pretty soggy napkin in Pocatello, Idaho in about 1986. And so that then led to this research relationship with Anheuser-Busch, and then we leveraged that for additional funding from the American Malt Barley Association, Great Western Malting, other players, where we were kind of like this little back eddy here in the world of barley breeding, where we were the folks working on winter 6-rows. And so it was a rewarding and, very successful, certainly from the genetic viewpoint, to be working on winter 6-row. But there was just little or no commercial adoption. And we had some resistance at that point, particularly from the emerging craft brewing industries and the craft malting industries to Sixrow, because these folks have been brought up in, if you will, a European tradition where they wanted tworow, and the idea of making a craft brew from Sixrow was analogous to eating human flesh. They just didn't want to mess with it, Now, things have changed radically. So, for example, a Sixrow winter type released from our program called the variety Alba has been very successfully malted by Skagit Malting up in Mount Vernon, Washington, and Pike has brewed some excellent beers out of it. So it goes to show that, if there's a will and there's a way, then, tworow, Sixrow, it can all work. So I switched over to tworow. Our program is now about 100% tworow on the malting side. It just simplifies things if you work on Sixrow or tworow. So we switched to tworow. And for us, it's kind of like having switched to the metric system finally after being in some far corner of the world where you worked with pounds and inches. Suddenly you're like the rest of the world. So I'm glad that we've made the switch, and it just kind of makes it easier to work with everybody: mainstream brewers, craft maltsters, mainstream maltsters.
Awesome. I thought I'd mention your website real quick, barleyworld.org. Throw that up there. And that's where you got a lot of your research posted, right?
Yeah, so we've got all kinds of research, findings posted there. We have data from our ongoing trials. We've got publications of one sort and another, descriptions of our varieties, and then some ongoing research projects that we'll refer to a little bit later in this conversation, such as the barley contributions to beer flavor project. But before we get there, maybe we wanted to focus on a couple of other traits, like low temperature tolerance, for example.
Yeah, that was what I was going to ask you next. Part of your research obviously focuses on temperature tolerance. How do you make a barley that really is more tolerant of cold?
Well, we should say, how do you try to make a barley that's really more tolerant of cold? And it comes back to that kind of genetics breeding interface, in that we want to understand the genes that make barley resistant to low temperatures. And once we understand what those genes are, then we should be able to do a better job of breeding for cold tolerance. I got to confess that I live in the Willamette Valley of Oregon, where it doesn't get that cold. And I've got colleagues who live in places where it gets awfully cold. So first, I have to have a little cred about working with winter barleys. One of my key collaborators is Kevin Smith at the University of Minnesota, and so it definitely gets certifiably cold in Minnesota, and so we do some of our testing there in collaboration with Kevin, and then we do the genetics here in Oregon. We also have some areas within our own state in the Pacific Northwest, on the east side of the Cascades, for example, where low temperature tolerance is needed. Now, you could ask the question, well, why do you even want to work on low temperature tolerance to even begin with, when we live at a time of global warming? So, yeah, the net trend is towards global warming. That certainly led to some water shortages, and it's going to lead to continued challenges for water. And so I think that's going to force us to look at barley that is more water use efficient. And kind of a shortcut to water use efficiency is to think about raising barley in new places and at different times of year than we've typically raised it. So if you can raise barley in an area that has a winter precipitation pattern, as we do in much of the northern hemisphere, say from the Pacific Northwest on into the upper Midwest, then you'll be able to raise your crop capturing the incident rainfall or snowfall, and not be relying on increasingly episodic summer rainfall or very expensive irrigation, where you're competing with higher value crops, with endangered species and so forth. So
maybe drought tolerant barley is what you're looking at as well?
Well, exactly. And we use low temperature tolerance as an indirect way to select for drought tolerance. Turns out that selecting for drought tolerance is really a challenge to properly control moisture levels within your experiments, whereas it is somewhat easier to test your barley selections in areas that do get cold and select those that survive.
So is it primarily through selection that you're doing this process, or are you also doing a lot of genetic work associated with it?
Well, we do both. Let me give you two examples there. We just finished up a piece of work that was a genetics project. It was kind of an international team involving folks in Japan and Australia and Hungary and here in the U.S. And we were looking in that case at the relationships of growth habit and low temperature tolerance in a set of genetic stocks that had been developed by a research group in Japan. And in collaboration with a colleague at UC Riverside, Maria Munoz, we aligned the genetic architecture of these stocks with the barley genome sequences and specifically identified which genes were present. And then with colleagues in Hungary, Ildikó Karsai, we measured what those the actual low temperature tolerance was of these lines. And then with Ben Travascus in Australia, we actually looked at the expression of these genes. And so the net effect of all of that was that we can really push what we call facultative barley, that you can afford to give up what's called a vernalization requirement and still maintain maximum cold tolerance. And so it was such a cool story, particularly because these genetic stocks had been shared with us by a colleague, Kaz Sato, in Japan, and they'd been created back in the 70s by his predecessors at his institute. And they had just done this incredible job of systematically crossing in without the benefit of any sort of molecular information, just very small genetic segments that harbored the target genes. So that was very much kind of a genetic sort of a take on low temperature tolerance. We have another project that's ongoing right now that involves both selection and genetics, and that's a project that involves looking at 1,000 barleys that we selected from around the world, contributed by colleagues who had an interest in some data to show that these things were cold tolerant. We fingerprinted them with over 9,000 molecular markers, and we've grown them at 16 locations around the world over a 2-year period. So that is one big Excel file. Yeah, as you can imagine, putting
That's
that
together.
That's
a big
project. Yeah. So we've got a student, Dustin Herb, who's using a technique called genome-wide association studies to put all of those what we call trait data, phenotype data, and the genetic data together to identify those regions of the genome that are responsible for low temperature tolerance. So exciting time.
So then once you understand that, you can cross the plants, hopefully, and get a new strain. Is that how it works?
Absolutely, because chances are that we'll find something that's low temperature tolerant in a genetic background that malts like a rock. And if we want to have something that's going to modify nicely and give us great beer flavors, then we'll need to make that cross and then select those that have the target flavors and the low temperature tolerance. And hopefully the genetic information will guide that breeding program.
Now, disease resistance is obviously very important as well. How do you develop a barley that's less susceptible to certain diseases?
Well, the same sort of logic that we kind of laid out there with the low temperature project, where you identify sources of genetic variation, you identify barleys that are resistant to whatever disease you're concerned about, and then you try to find the genes that drive that, you select for resistance, and then you can use the information about the underlying genetics to guide your breeding program. For example, here in the Pacific Northwest, stripe rust is a fungal pathogen that we're quite concerned with. And we started working with a group in Mexico over 30 years ago to identify sources of stripe rust resistance and have been kind of systematically cataloging those disease resistance genes over time and then ensuring that they're present within our varieties. Climate change is just going to make it increasingly likely that we all face diseases that we just never expected to see in our growing areas. And so the more quickly you can respond to a new disease threat, the better off you are. If you're a conventional grower and your disease is fungal in nature, you have what are called fungicides. And so you can protect your crop with a fungicide. That's going to cost more to the grower. There's always unexpected environmental consequences with various chemicals, and then you also have the situation that the pathogen can evolve resistance to whatever you're applying. So whenever it's possible, we'd like to have varieties that are resistant to the diseases and possess what we call durable resistance, that is resistance that will last over time.
Very cool. Are you also, we talked a little bit about drought resistance, but are you also focusing on characteristics like per acre yield, for example?
Well, you have to, and our field experiments are very much designed to gear up and measure yield. I've got colleagues here at OSU, Scott Fisk runs our yield program, and when we sit down and design the trials that we planted just this last week, it was all about measuring grain yield on these things. And so we put out plots, we use experimental designs, we harvest those things with small grain drills, we plant them with small grain drills, harvest them with plot combines. And the goal there is to measure yield as accurately and precisely as possible in order to select potential varieties that will increase yield performance. So you might well ask, well, what about an heirloom variety out there? What if there was something that had exceptional flavor but didn't have the yield? I think we need to provide both maltsters, brewers, consumers with varieties like that, but the farmer needs to know what they're getting into. And whenever possible, they'd like to have a variety that's going to maximize production for them and reduce input costs. So yield is very much at the top of the list. Then after that, it's up for the maltster and the brewer to decide if they're willing to pay a premium to get a grower to produce a variety that yields less than some other options.
What are some of the actual methods you use to change the characteristics of the plant? Is the plant actually modified or bred to have better features? Is it primarily a crossbreeding program? How do you do it?
Yeah, we use crossbreeding. It's a pretty time-honored sort of tradition, and you can kind of break the breeding process down into a couple of kind of simple steps. And one is generating genetic variation. The second is testing the things you've created and selecting those that you want to carry on. And then the third is multiplying seed and assessing the commercial viability of what you've created. And so making a cross between two parents is a time-honored tradition. Mm-hmm. The typical cross would be high-yielding, poor malt quality, crossed by lower-yielding, excellent malt quality. Goal to select high yield, high malt quality. So as to how you actually measure those traits, that's where you start bringing in the whole breeding and genetics toolkit. Simplest thing is you put them out in the field and you look at them. Some things you can't look at and measure very well, like grain yield or malting quality, and so you actually need to measure it. Grain yield, we estimate, costs us about $60 per plot to measure on a 5-foot by 20-foot plot. A malting quality assessment at this point costs about $350.
Wow, that's pretty significant, yeah.
Yep. So the whole breeding process is one then that you've got some alternatives along the way. So if you're generating genetic variation, you could also do that by mutation. You could do that using a GMO technique. So those are some different ways to make mutation, but you're still going to have to test whatever you created out in the field. And so when people say that GMO is a shortcut and it's going to make breeding obsolete, I kind of would like to engage them in a bit more dialogue, because you really are going to have to test those GMO products extensively under field conditions to make sure that you perform as you want them to. And then even once you've selected something, then you're in the process of moving beyond the handful or the kilogram of seed that we may typically have in a breeding program up to the point where a farmer is going to be able to start assessing those things. And you figure it takes about 100 pounds of barley seed to plant an acre. And for a commercial maltster, for example, to gear up and test a potential selection, they need 350,000 pounds of barley. So the scales move up very quickly, and so you really need to think of that kind of long-term prospect for envisioning, designing your breeding program, and then getting that into the marketplace.
So do you have a lot of test plots at the university? How does that work?
Yeah, so we have university farms. Every university has a farm, and then they have what are called experiment stations around the state, and those provide places where you can conduct what we call small plot research. And so, yeah, like you're showing at the website there, those are some of our smaller plots. As I said, the typical yield trial plot might be about 5 feet by 20 feet, and you might have 2 or 3 replications of that. We'd also like to do as many on-farm trials as we can. And so just right now, for example, we collaborate with 2 farmers here in the state of Oregon, the Herb family here in the Willamette Valley, and then the Klon family over in the Madras area. Those are pictures of small plots being harvested there. Then as we move up onto the on-farm trials, we can continue to use our own small plot equipment there, or as soon as we generate larger volumes of seed, we can look at doing maybe at least an acre with the farmer. Their equipment is of a scale that still they can barely get in and turn around on an acre. They're going to need more than that typically.
So, I mean, what impact does your work have on things like harvesting, drying, and malting the barley?
Well, certainly those are just critical concerns for the grower, the maltster, and ultimately the consumer in terms of sourcing the beer that they expect. So in terms of harvesting, one thing is obviously we target the harvest date. We want to have varieties that mature at an optimum time for a given growing area. We can also select for things like resistance to scalping or skinning, and so you want a malt barley to have an intact hull. You want resistance to breaking in the combine, so you want a fairly robust seed there. Drying, we do select for things that relate to moisture at harvest. One would be pre-harvest sprouting, which is when you have untimely rains. You'd like to have just the right amount of dormancy in your crop to resist the untimely rain, but at the same time not cause problems for the maltster. And then malting. Oh my, that's just the exciting place where we are right now. And you were just showing some pictures of our mini malting system here at OSU.
So you got your own Malthouse there, it looks like.
Well, we have the beginnings of a Malthouse, and we're optimistic that we'll have some stocking stuffers of Oregon State University malt by Christmas. So the unit that you were looking at is what we call the OSU Mini Malter, and that was a unit that was designed by engineering students here at OSU and was designed to bridge the gap between the Barley Breeding program and the Fermentation Science program that we have, because historically, I'm breeding varieties over in my building. Tom Shellhammer and his colleagues over in the Fermentation Science are brewing barleys, but there was no way to make malt from our experimental selections at a small enough level that we could get them into the pilot plant. So we designed with the engineering students this unit that will do about 100 to 300 pounds of barley, and that's enough to feed the pilot system that they have in Fermentation Science. So if we have a potential new variety, or we want to grow a variety in our area and see what it does for beer quality, we'd be able to quickly do that and say, invite in a local panel of brewers and consumers and say, hey, what do you think of this beer? There's just no way that we could do that in the past.
And it looks like you got your own brewing system as well.
Yeah, so the Fermentation Science program here, now the pictures you're looking at right there, those were taken up at Rogue, the Rogue facility up at the Green Dragon in Portland, but they were using one of our experimental malts in that particular brewing system. And in that series of pictures, the bottom picture that you just showed there was the facility that we've just remodeled. This was before remodeling, where we have our malt lab. And so that's all going to be up and running, and we'll as well have a facility for doing analysis of our malts. And a lot of that was a fundraising project and some of the number of key donors there that are mentioned at the Barley World website. And I'd also like to just throw in a pitch for the Brewers Association, which put in a significant chunk of money to that project.
Fantastic. Well, what are some of the implications for the new barley varieties that you're developing for beer brewers?
Yeah, so we have, I'd say, kind of two lines of research that are going on there. One would be the mainstream barley development effort, and so there we're targeting two-row, low temperature tolerant, winter and facultative types for both the adjunct brewer and then the whole malt brewer. And we have two new selections that have just come into that are modestly called ORW1 and ORW2. If we want to be optimistic, the code names are Thunder and Lightning. So, we'll kind of see if they hold up.
Nice.
So those are two in the system, and then we have a whole pipeline full of exciting new potential varieties. We have another research endeavor that's kind of a smaller component, a very focused one within our program, and it's called the Barley Contributions to Beer Flavor Project. And that's funded by a consortium of 6 craft brewers we call them the Flavor Six Pack. And there we're asking the kind of audacious question, does barley actually contribute to beer flavor? And so that's opened up this whole new line of research for us where, clearly hops contribute to beer flavor, and that's led in large part to the explosion in the craft brewing industry. We know that yeasts and some of yeast relatives contribute huge flavors, and that's led to the whole exploration of Belgians and sours and saisons and so forth. And even water, we all appreciate the flavor components. So in barley, we're kind of pushing this flavor thing with full recognition to the maltster's art. Because without a doubt, the maltster takes the same barley and they can make everything from a pale malt to a roasted out of that same barley. So the malting process brings a whole palette of colors and flavors and so forth. But we'd like to ask the question, are there some undiscovered flavors out there in barley that perhaps might first fit into some of the lighter colored malts and perhaps the spectrum of beers from lagers, pilsners, and up to ambers, but maybe those flavors could also interact with hops and yeast in unexpected ways. Very
cool. How long does it take a new variety and actually get it commercialized? Obviously, you're doing pretty small plots. It must take a while, right?
Well, it does. Thankfully, barley is a very productive sort of a plant, and so you have a nice multiplier there. Off of one of those small plots, we'll get several kilograms of grain, and so if you took those and multiplied them one more round, you could be up to an acre increase the following year. You can use tricks of increasing seed in the southern hemisphere for us northern hemisphere breeders. So there's a lot of ways you can kind of blow up the amount of seed. But if you look at the conventional route for barley varieties, it's a 10 to 15 year process to get from grain to glass. And I would say more like the 15-year process. Of that, probably 5 years is spent in the plant breeding side of things, the plant breeder's black box. The other 5 to 10 years are spent in the kind of maltster and brewer black box, because in general, they're going to want to take a deep look at a potential new variety and ensure that it's going to fit their malting and brewing schemes. Probably the biggest news on the horizon for barley breeders in recent years would be the development of a craft malting industry, because the craft maltsters are able to malt in smaller volumes than the larger maltsters, and so that gives them greater flexibility to look at other varieties, and then they can move those in smaller amounts into the craft brewing sector. And so the variety interest and adoption rate, I think, will really accelerate as you have more craft brewers coming on the market. Potentially, they could see something that came out of our breeding program in a 4 to 5 year period and say, hey, I want that, and move immediately into a craft malting trial and have that in a beer within 6 to 7 years. Very
cool. Somebody told me, I don't know if this is true, that there's a relatively small number of barleys are used today in commercial production. Is that true for beer producing?
Absolutely. The U.S. has historically had probably one of the slowest variety change rates in the world. And that's been due to the fact that barley prices are relatively low. It's been a high volume, low margin commodity sort of an item in the business out there. And so maltsters were reluctant to change varieties. Brewers were comfortable with what they had. And the increments in yield performance were fairly small and really not enough to force anybody to change anything. There was a little different picture in Europe. They had a much quicker turnaround in their variety adoption rate. And so, again, I think a lot of that's going to change as we see some changes in practice in the malting and brewing industries. And already we've seen that with a couple of new varieties moving through the American Malt Barley Association approval process much more quickly. You had the variety Went Malt, which was a European variety that had already been tested in Europe, but it was approved immediately for production in the U.S. based on its European track record. You had Thoroughbred, which is a Virginia Tech variety that was approved because of interest of craft maltsters. You've had the variety Full Pint out of our program, which is getting a bit of traction on account of its flavor, and it's not AMBA approved yet, but if it moves on into sufficient scale, there's no reason that wouldn't happen as well.
What can brewers look forward to as some of these new barley types come online?
Well, I think it's just going to make their life more complicated because they're just going to have to make good choices, and it's going to be so hard to decide which one of these exciting new barleys they want to work on. We're fortunate in barley that we have not had any sort of kill-alls, like some disease that comes in and just ravages the barley crop to the point that the maltster and brewer have to change variety. And so up to now, it's been kind of their choice. I think with some of the new flavors that are coming on, they're going to say, wow, do we want to avail ourselves of these? We've been working with New Glarus, and you can see some of the data posted at Barley World in the Barley Contributions to Beer Flavor area, but they're discovering some new flavors in some of these experimental types that we've had coming out of crosses of Full Pint by Golden Promise, for example. And so there'll be kind of a race to get those commercialized and for people to try those in their beers. There's a whole other type of barley that I think is really going to be worth having a look at, and those are naked barleys where the hull does not adhere. And typically those have been used for culinary, human nutrition sorts of barleys. But with some advances in brewing technology, such as the use of mash filters, the role of the hull in filtration is perhaps not quite what it was. There could just be some crazy undiscovered flavors in some of these things.
So you're saying you get actually a hull-less barley from that to take out the astringency, I guess? Yeah.
Exactly. So just imagine that you could have a Reinheitsgebot compliant Hefeweizen. So my German skills are limited enough, so it wouldn't be a Hefeweizen. I think it would be a Gerste Hefe or something, since there's no Weizen in it. But it could be made from all barley malt, from an all naked barley with hops, yeast, water, and that would be it.
Opens up a lot of possibilities with the roast barleys too, because obviously as you get into especially you get into the 100, 200 range of Lebobon, you get a lot of astringency that comes through from the hulls, right?
Exactly. And I was just at the Master Brewers Conference in Jacksonville and was visiting about that with a couple of maltsters. And so we also work on naked barleys, and we shipped out some samples to some of them to have a look at for just those sorts of uses.
Well, Pat, I wanted to ask, is there anything else you'd like to add?
Well, Barley World is out there, so for any of the listeners, viewers that would like to learn more, please have a look at it. Feel free to contact us.
Yeah, it's barleyworld.org for those of you listening on the podcast.
Barleyworld.org. We try to hold a Barley Day every year in the spring, so come out and see us. That'll be announced at Barley World. And just sort of more generally, I'd encourage all of you out there to kind of work through the glass to grain system backwards. Get to know your local brewer. You probably already do, but get to know your local maltster if you don't already. Move back through the chain a little bit and get to know your local barley farmer, and then go discover who's breeding barley for your area.
Well, Pat, really appreciate you coming on the show. Thanks so much for being here.
Brad, it's really been fun, and look forward to working with you in the future.
And my guest today was Dr. Pat Hayes. He's professor of barley breeding and products at Oregon State University. He specializes in genetics and breeding of new barley products. Well, thanks again to Pat Hayes for joining me this week. Thanks also to Craft Beer and Brewing Magazine, where you can get your 15% discount on their magazine or anything in their store if you use the offer code BeerSmith2015 when you shop at beerandbrewing.com. Again, use the offer code BeerSmith2015 and get your subscription now at beerandbrewing.com. And also, BeerSmith software, the industry standard for home and professional brewers that lets you design great beer recipes and brew with confidence. Download your free 21-day trial today from beersmith.com. Thank you for listening. I hope you have a great brewing week.